Hello everyone and welcome to today's seminar. My name is Hazi Patel and I'm your host for the event. Today's webinar is entitled A Compressed Genome-wide Perturb-seq Screen to Dissect the Polarization of THP1-derived macrophages. And before I introduce our presenter today, I'd like to encourage you to submit questions which we'll address in the Q&A session that follows the presentation. So feel free to submit questions throughout the presentation. We will answer them as text, but we'll also take some questions in the Q&A session. So with that, I have the great pleasure today of introducing our speaker, Tilman Bergstürmer. And as background, Tilman is a CRISPR enthusiast since the early days of CRISPR. He originally trained as a biochemist. He joined Haplogen as a principal scientist and then later became their CSO. Following the acquisition by Horizon Discovery, Tilman served as director of R&D and later head of innovation, where he oversaw Horizon's technology platform and innovation agenda. And then in 2008, he co-founded Milea Biotechnology, which focuses on single-cell CRISPR screens. And he's also the CEO of BitBio Discovery, a joint venture between Vienna-based Milea Biotechnology and Cambridge-based BitBio. Tilman is passionate about science and he enjoys working with multidisciplinary and multinational teams. And with that, Tilman, I'd like to hand over to you.
Brilliant. Thanks for the intro, Hattie. Again, my name's Tilman Bergstürmer. I'm the chief scientist of Milea. And at Milea, we focus on single-cell CRISPR screens. And today, I'll present sort of a very exciting example, a collaboration we've had with BD Biosciences on a compressed genome-wide perturb-seq screen to look at THP1-derived macrophage biology. Now, to start, CRISPR screens sort of come in two flavors. They can either be pooled or arrayed screens. In a pooled screen, we start with a population of cells that grow in a dish, and we introduce multiple guide RNAs as a guide RNA library so that every cell in the dish receives a different guide and sort of becomes a knockout for a different gene. And then we're querying a certain biological question in a pooled fashion. And often these screens are focused on cell fitness, cell growth, and cell proliferation. The converse screening paradigm is one that is referred to as the arrayed screening paradigm, where we grow cells in an arrayed format, let's say in a 96-well plate, and every well receives a different guide RNA. And so as a consequence, we can then assess the phenotype that is sort of changing as a consequence of the genetic perturbation in the arrayed format. And this could really be linked to any phenotype of your liking. Now, the pooled screens have the big advantage that they're easily scalable, and that's very easy to understand because you essentially grow cells in a big dish, and to make that dish a little bigger and go to genome scale is sort of a very easy task. The downside of pooled screens so far has been that they're often linked to rather simplistic readouts. And the converse screening paradigm, the arrayed screening paradigm, is great because you can actually be linked to any readout of your liking. You can look at cell supernatants and assess a cytokine. You could image cells. You could analyze their transcriptomes. But the downside is that they're difficult to scale. And so there is sort of an emerging group or an emerging class of CRISPR screens, which are pooled screens, yet they're linked to an information-rich readout. And one of the variations of that theme is a screen in which CRISPR perturbations are linked to a flow cytometry readout, where you sort cells into different bins based on a certain cell surface marker or based on a reporter. There is a second flavor, which is illustrated on the bottom, where you do CRISPR perturbation on plates and you assess the phenotype by imaging and essentially then identify the guide RNAs through in situ sequencing or by some other means. The third flavor is the one that we're focusing on today, and that is to combine CRISPR perturbation with single-cell RNA sequencing. And the way to do this is we we separate cells out on a grid, and on that grid, every cell ends up in a different well. We put beads onto these wells as well, and the beads contain barcoded oligos that allow reverse transcription on the surface of the bead, thereby creating a single cell transcriptome. And again, that work presented here today was recorded with the BD Rhapsody instrument, more specifically with a novel version of it, which is called the Xpress, that is compatible with higher throughput. So if we now sort of zoom in one level, the experimental workflow that we're interested in, again, is a pooled screen. So we start with a cell that already expresses Cas9, or in some other instances, we bring sort of Cas9 in exogenously. We then deliver a pooled guide RNA library by lentiviral infection, so that every cell in a dish is a knockout for a different gene. And then we perform single-cell RNA sequencing. We single cells out in the instrument, in this particular case, the BD Rhapsody. We transcribe the transcriptome, or reverse transcribe the transcriptome on the surface of the beads, and then sort of throw everything together and, based on molecular barcodes, identify which transcript belong to which cell. And sort of even more importantly, for that specific technique presented here, link the transcriptomes that we sequence by single-cell RNA sequencing to the guide RNAs that elicited the perturbation. And that workflow was first sort of pioneered in the lab of the Milia co-founder Christoph Bock. That's the work sort of cited here, but there were parallel efforts in other labs as well. So what are the key applications of CROP-seq, perturb-seq type approaches? I would say most of our customers have an interest in target identification or target validation. That's still the sort of number one question: identifying a relevant model perturbing a set of genes and identifying a target whose knockout is sort of impactful with regards to a particular phenotype. The second set of questions, if you like, has to do with epigenetic editing. So we've been able to develop a workflow to perturb enhancers and connect enhancers or SNPs to the genes that are regulated in terms of expression level by these enhancers. And the third area is really around small molecule mechanism of action. So we combine CRISPR perturbation with a drug treatment, and we ask sort of how do the two interact, and can we identify gene knockouts that render the cells particularly sensitive or particularly resistant to a drug treatment. Now, in order to lay the foundation for today's experiment, I would first like to have a little branch out on targeted single-cell RNA sequencing. This is a particular technology that we're very excited about, that was actually also pioneered by BD Biosciences. To illustrate the problem, we took data from a very conventional whole transcriptome single-cell RNA sequencing experiment, and we plotted the 20,000 genes on this plot. And so on this plot, we're plotting on the x-axis the expression level of a gene, and on the y-axis the variance. And what you can appreciate is that in that particular experiment, again, every dot is a single human gene or a single human mRNA. Very few mRNAs actually change across the cells that were analyzed in that experiment. And so only the ones that change, only the genes that change, provide actually information as to how the cells differ from one another. So those are the genes that would actually like to sequence. However, in sort of conventional RNA sequencing workflows, we're sequencing all the 20,000 human genes. So what we would ideally like to be able to do is zoom in and focus on that boxed set of set of genes and ask the question, can we amplify those selectively? And we developed an in-house protocol that is very similar to the protocol that is used at BD. That was based on a publication. The protocol we developed was based on a publication from EMBL from 2020, where they came up with a way of amplifying selected mRNA. So the first step of the single-cell workflow is very similar to conventional single-cell RNA sequencing. Transcripts are amplified on the surface of beads, and molecular barcodes are included that sort of mark the transcripts. But then in a second step, we design a multiplex primer panel in order to amplify selected mRNAs that we would like to keep and throw away the rest. And the first advantage of this is that we can get away with a lot less reads during next-gen sequencing. And that's a significant advantage because the screens and the experiments that are based on single-cell RNA sequencing are still very costly, and one huge cost component is next-gen sequencing. So in this particular case, we were able to reduce the number of NGS reads from 50,000 reads per cell in whole transcriptome sequencing to 10,000 reads per cell in targeted sequencing. Now, how deep can you go? That's a question that we often get with targeted sequencing. And in order to address this, we took a cell line of which we knew this is a cancer cell line, of which we had recorded the transcriptome by bulk RNA sequencing. And that cell line expressed around 11,700 genes. And now we put these 11,700 genes into different quantiles, where the tier 7 quantile is sort of represents the genes that are very abundant, where the tier 1 quantile represents the genes that are sort of very close to not being expressed. And from these 7 quantiles, we picked 35 genes each. And we either sort of identified them by whole transcriptome sequencing, sort of a posteriori after the experiment, or we designed primers for those 7 times 35, so 250 or so genes, and amplified them by targeted sequencing. And then again, the targeted sequencing experiment was sequenced at 10,000 reads per cell, while whole transcriptome sequencing experiment was sequenced at 50,000 reads per cell. And what you can immediately appreciate here is that all the dots on this plot cluster on the lower right of the plot. So here we're asking, in what percentage of all cells do we detect a given transcript? And you see that for the tier 7 genes, there's not that much of a difference. They're really detected well with both techniques, both whole transcriptome and targeted sequencing. But for all other transcripts, and particularly for those transcripts that are lowly abundant, we really only see them well or see them at all in targeted sequencing. So we're not only saving money here, we're actually increasing depth, which we find very advantageous. And so as a next step, we performed a little CROP-seq screen. Now in this screen, we've perturbed a bunch of genes. This is the set of genes listed here on the x-axis. We stimulated cells with interferon, so that the pathway or the biology we're querying here is the JAK-STAT signaling pathway. We stimulated these cells with interferon beta and asked what are the genes that impact on this pathway. And so if you just compare the two heat maps here, the one derived from whole transcriptome sequencing and one derived from targeted sequencing, you'll immediately notice that the signal to noise ratio in that experiment is so much better in targeted sequencing. In whole transcriptome sequencing, you can kind of guess that it's that set of 6 genes on the left that have a phenotype, but that is sort of very clear in targeted sequencing. So again, just to summarize that part, we're very excited about targeted sequencing because it increases the depth of the experiment. So it sort of provides more information on the set of genes that you're really interested in, yet reduces the cost base significantly. Now, what's the model system that we chose for the experiment presented today? We wanted to study macrophage biology in that experiment. And more specifically, we wanted to study the polarization of macrophages. So macrophages are part of the innate immune system. This is the immune system that is distinct from the adaptive immune system. Adaptive immune system is composed of B and T cells, and they're very specific, if you like, towards the attacker, towards the invading pathogen. Whereas macrophages have sort of more generic defense mechanisms that can be active against a variety of different pathogenic species. And these macrophages come in at least 3 flavors. Sort of initially, sort of when they develop from monocytes, they're qualified as M0 macrophages, sort of not quite specialized. But then they have at least two ways of specializing, and one is called the M1 macrophage, and the other one is the M2 macrophage. Now, in terms of function, you can imagine that in a situation of an infection, the body first wants to create a pro-inflammatory environment to battle the infection, and that's the task of the M1 macrophage. But then, once you've eliminated the invading pathogen, you want to create a scenario where the tissue is repaired and where the damage is sort of removed, and that's the task of the M2 macrophages. So it's very important for the immune system to be able to tune the function of the macrophage and sort of either convert them more into an M1 or more into an M2 macrophage. And this actually has therapeutic potential or therapeutic implications. So in a tumor situation, we actually want to sort of promote M1 macrophages because they, in terms of function, have the function to battle the tumor, whereas the presence of a lot of M2 macrophages is actually not good because that suggests that the immune system is sort of not alerted to the tumor. So that's the biology we're studying. We turned our attention for the particular screen I'll present in a moment towards a model system. And the model system that we have used here and others have used beforehand is a THP1 cell line. And that is a monocyte cell line. It looks like this sort of originally. So these are suspension cells. But they can be treated with an agent called PMA, and that agent converts them from suspension to adherent cells. So they become macrophage-like cells, I should probably rather say, similar to the M0 macrophage, not yet sort of decided whether they want to become the pro-inflammatory or the anti-inflammatory macrophage. And then we can actually provide them with a little cocktail to skew them into either direction, M1 or M2. And the cocktail we use for M1 is LPS and interferon gamma, and the cocktail we use for M2 macrophage is IL-4 and IL-13. And we did that with a specific model here with the THP1 cells, and we recorded the transcriptome by bulk RNA sequencing and identified a set of genes that mark the M1 or the M2 state. And that's important because this is sort of the biological process we want to study. And those marker genes will be utilized to identify genes that regulate that transition between M1 and M2. As a next step, we created a cell line, a stable cell line, in which we integrated dCas9 fused to CRAB. Now, dCas9 is the active ingredient of the CRISPR system. In this particular case, it's a catalytically inactive version of the enzyme that is fused to a transcriptional repressor. And so if you recruit the system to the transcriptional start site of a gene, it will transcriptionally downregulate that gene. So it will downregulate transcription of the gene so that you create a knockdown for that particular gene. So we're not using the sort of CRISPR nuclease in this particular screen, but we're using CRISPR interference, and I'll come to that in a moment why that is sort of an advantage in this particular experimental setup. Now, what we then did is we introduced two specific guides into the cells in order to see if the system was functional. And the one guide that is a control point or reference point in the experiment is a non-targeting guide that is marked NTC. And the second guide targets a cell surface marker called B2M. And B2M is the advantage that we can read out the levels of B2M at a single cell level by flow cytometry. And you'll appreciate here that the non-targeting guide doesn't do much to B2M levels. This is the y-axis of the FACS plot, whereas the B2M guide leads to an almost complete knockdown of B2M. So it says the system is functional, we can deliver a guide, and we get a knockdown for a given gene of interest. Now, what does the screen look like? Here in this particular setup, we wanted to utilize the new instrument, the BD Rhapsody HD Xpress, and really perform a screen of unprecedented scale. And in order to do that, we turned our attention to a novel screening paradigm that Brian Cleary and others have called the compressed perturb-seq paradigm or compressed CROP-seq paradigm. Now, what does that mean? If you were to run a CRISPR screen where you deliver a single guide RNA per cell with a genome scale library, you would have to screen an enormous number of cells by single-cell RNA sequencing. And we calculated that that number is on the order of magnitude of 9 million cells sequenced by single-cell RNA sequencing. So that would really break the bank. And so we thought, can we come up with a trick? And the trick we came up with is we try to deliver multiple guide RNAs per cell. So every cell does not have a single guide RNA, but has multiple guide RNAs in that cell. And obviously that only makes sense under the assumption that few guide RNAs will do something to cells. If a lot of the guide RNAs that we did something, that we applied did something to the cells, then we would get a huge slim muzzle that we could not deconvolve. But here, that assumption was sort of valid. But then introducing multiple guide RNAs into one cell has another problem, has a biological problem. And the problem is that if you were to do this with Cas9 nuclease, the cells would just die because you elicit multiple DNA double-strand breaks in a cell, and cells don't tolerate DNA double-strand breaks. As sort of biological cells, they've learned if there's multiple DNA double-strand breaks in a cell, I should die. And so we had to turn our attention to CRISPR interference, because CRISPR interference does not introduce any DNA double-strand breaks. It utilizes the inactive version of the Cas9 nuclease. And as a consequence, we could actually introduce multiple guide RNAs per cell and see the consequences in the screen. And that allowed us to squeeze the entire genome scale library into a little less than 1 million cells per experiment. So it's a trick in which we can squeeze a lot of perturbations into a small number of cells. That's why it's called a compressed screen. Now, this is what the experiment looked like. We started off with a THP1 cell that already harbored dCas9 fused to the transcriptional repressor CRAB. We introduced a genome-wide pooled guide RNA library. I'll speak to that in a moment. And we introduced it at high multiplicity of infection. This means that multiple guide RNAs land in every cell. We then eliminated some of the cells that had not been transduced. There were very few. And then performed single-cell sequencing using the BD Rhapsody instrument. Most of the cells that we sequenced were in the M1 stage, but we included a few monocytes and a few M0 macrophages as reference points in the experiment. We also included a site-seq or app-seq panel. This is a panel of antibodies that stain specific cell surface markers. And because these antibodies have been linked to DNA barcodes, they can be read and interpreted by single-cell RNA sequencing. We then performed single-cell RNA sequencing, capturing both the RNA and the antibody-based barcodes, and at the end did the computational biology analysis. What is the library that we used? The library that we used is a CRISPR-I library that we designed in-house. It was designed sort of incorporating several features. A key feature of every guide RNA is the sequence of the guide RNA in the surrounding space. But our design algorithm also includes epigenetic data as well as positional data, sort of how close is the guide RNA to the annotated transcriptional start site. And the library we designed is a genome scale library, so it targets around 19,000 human genes. And we compared the performance of the library on a set of sort of published data points. And we think that it's sort of at least on par, possibly superior to some of the published guide RNA design algorithms, particularly, I would say, for CRISPR interference. Now, I mentioned previously that we wanted to utilize targeted sequencing because it, on the one hand, reduces cost. On the other hand, it allows us to zoom in on specific processes and capture these with great depth. And so we designed a primer panel that focuses on 357 marker genes. And I would say a huge chunk of that panel was informed by the bulk RNA sequencing experiment that I described earlier that distinguished M0s, M1s, and M2 macrophages. This is sort of the large chunk. But then it also included the genes that we're measuring by site-seq, app-seq. And the reason is we wanted to utilize that experiment to compare how well RNA levels and protein levels sort of compare or match each other. We then included a bit of published markers from the literature. And then we sort of included little mini panels of 20 to 30 genes which focused on different biological processes. And the beauty here is because we have a genome scale library, we're really expecting all of these processes to be affected by some of the gene knockouts. So it's really an inverted, if you like, it's an inverted screening paradigm. In the earlier days, we used to select a small number of genes to perturb, and we looked at all the transcripts of the human cell. Here we're doing the opposite. We're perturbing all the genes, and we're just picking areas of biology for which we design marker sets, and we sequence those selectively by targeted sequencing. Let's look at the quality control of the experiment. Again, the experiment was conducted on the BD Rhapsody HT Xpress. We did targeted NGS, or sort of targeted library prep and NGS. The first sort of key metric down here shows the distribution of guide RNAs across cells. And again, keep in mind, we try to bombard these cells with a high dose of lentivirus so that every cell on average would have multiple guide RNAs. And in this particular experiment, we ended up having around 5 guide RNAs per cell. We loaded around 1.6 million cells on the instrument. We retrieved around 930,000. And then following sort of elimination of some of the doublets, following elimination of some of the cells that had to be removed sort of for quality reasons, we ended up with 460,000 cells in which we could call at least one guide RNA. And then with 5 guide RNAs on average, we ended up having around 160 cells per knockdown in that experiment. And keep in mind that entire experiment was done with sort of 1.5 million cells, and it was sequenced on a half NovaSeq X experiment. So it's really a screen, a very large scale screen that is sequenced with very limited resources and depth. Now, the first question you could ask is, do we find the 3 cell types that we included in the experiment? Keep in mind, we spiked in these monocytes and macrophages of the M0 type and mainly had M1 macrophages. And lo and behold, we can distinguish them based on their transcriptomes. This is sort of comforting, but maybe not a great surprise because these cells are quite distinct from one another, and we had determined their transcriptomic identity by bulk RNA sequencing previously. Next question is sort of how do we call guide RNAs? And this is obviously a little bit difficult in this particular setup of where sort of cells have multiple guide RNAs integrated. And so what we essentially do then is we just look at that blue guide RNA here as an example, and we circle all the cells that have that particular guide RNA. We ignore the fact, at least for the time being, that these cells have other guide RNAs, and we compare that set of cells with a blue guide RNA to a reference set that has sort of unrelated guides and not the blue ones. So by doing those comparisons, we can now analyze what happens to the cells that have the blue guide RNA. And that is, of course, the key question we're trying to ask. Not the blue sort of specifically, but sort of per guide RNA, try to resolve perturbation phenotypes. And so the first phenotype we looked at was, do we see the knockdown of the 300 target genes? Now, we can only see the knockdown for the genes that were included in the targeted primer panel. And that, again, was a set of 357 mRNAs. And so we plotted the CRISPR-I perturbations on the x-axis against the cognate mRNA levels. And what you can appreciate here is that that plot shows a diagonal. And the diagonal indicates that we see a knockdown for the cognate gene at the transcriptomic level. We see that knockdown significantly in sort of 238 out of 357 genes. So that's, I would say, that's great. Now you wonder why, there's still a good third of the genes for which we're not sure whether we have a significant knockdown. And one of the confounding factors here is that despite the fact that we're using targeted sequencing, some of the genes that we targeted were lowly expressed. and they had a TPM of less than 5. And so the worry is that maybe they're too lowly expressed in order to see the downregulation. So we cannot distinguish between them being lowly expressed and the low expression level being even sort of slightly decreased following the CRISPR perturbation. So lo and behold, to us, this plot would say that the knockdown worked in that experiment, and we're able to call guides as shown on the previous slide. Next slide is probably the key slide, or one of the key slides. So the question is, what genes and how many genes do have an impact on macrophage polarization? And so in order to determine that, we applied a computational approach that is called linear discriminant analysis. And so essentially, we look at the M1 and M0 macrophages, we compare them to another, and this method creates, if you like, a ruler that compares the two states. And we can then ask what are sort of we can create an LDA score, and we can ask what are the gene knockouts that shift the balance between the two cell types. A knockout that does not do anything is sort of probably sits somewhere in the middle between the two types, but one that influences cells to become more or less M1-like would shift the balance. And we came up with a set of genes shown on this plot that some of them quite significantly sort of affected M1 polarization. And those of you who are macrophage biologists will immediately appreciate that some of those genes make a lot of sense. Now, you need to keep in mind that these M1 macrophages are created by stimulating cells with LPS and interferon gamma. And the receptor for LPS is toll-like receptor 4, and the receptor for interferon gamma is IFNGR or IFNGR1. And obviously, the knockout for those genes, you would expect to have sort of a very significant effect on that phenotype, and that's what we saw. We also uncovered a bunch of signaling mediators, most notably JAK1 and TYK2. Those are two kinases associated with interferon gamma signaling, and if you knock them out, interferon gamma signaling can no longer happen. And then we found a few, let's say, less well-established but still intriguing genes that had been implicated in macrophage polarization or macrophage biology, I should rather say, and this includes UBE2L6, LAMTO4, and SPI1. And for some of these, we see actually very strong phenotypes. This is shown on this particular plot. So again, this is the UMAP representation of the entire experiment. So it shows the greatest degree of variation in the experiment. And again, these two knockouts for TLR4 and IFNGR1 lead to a very strong clustering of cells in a particular area of the plot, which again says these have very strong phenotypes on M1 polarization and on macrophage biology. Now, again, for us, the key question at this point was not so much, do we now uncover fantastic novel biology? The key question at this point was, can we sort of recapitulate biology that is actually meaningful? And I would say, based on these observations, we're quite happy with how the screen went. I want to address sort of two more questions before I close. The first one has to do with a comparison of the mRNA and the antibody profiles that were captured by Rhapsody. And so this is plotted here for a particular subset of genes. Now, here we're focusing on the genes that are part of the AppSeq SiteSeq panel. This is a set of cell surface markers for which antibodies were added. And we compare how they differ between M1 and M0 stage. This is the middle column. And then, in those instances, if they're upregulated in that transition, we give them a red color, whereas the downregulation is indicated by a blue color. And what you can immediately appreciate is that mostly these colors match, meaning if an mRNA, if a gene is upregulated at the mRNA level, as judged from the targeted mRNA sequencing data, is also upregulated at the protein level. This is reported by the SiteSeq column. And lo and behold, if you look at two examples here on the right, I would say that the profiles of mRNA and protein overlap reasonably well. There are some instances where the protein levels are sort of change more strongly than the RNA levels, but that's expected. It's also fair to say that the changes we observe are in line with what we expect. So we included some of the arrows here on the left to highlight what is expected in that transition. In other words, HLA-DR is a marker for M1 cells, and so we expect this to go up quite significantly, and that's what we saw. And conversely, CD33 is expected to go down, and we also see that on with CD33, we actually see it stronger at the mRNA than at the protein level. So again, CROP-seq is a useful addition to the toolkit. We love it, particularly with immune cells where there's sort of curated antibody panels out there, and we try to include them whenever we can. The other piece I wanted to share is a piece on a different biological process. Now, if you recall our panel, most of the panel was focused on macrophage biology, but there were some other islands of biology that we also queried by looking at mRNAs, of sets of mRNAs in that primer panel. And we wanted to see, did any of those panels report anything interesting? And the one example I wanted to highlight as part of that presentation is a panel of mRNAs that have to do with cholesterol biosynthesis and cholesterol uptake. Now, this is sort of the set of genes that were included in the panel. And what is known from the literature is that that process is actually regulated in cells, and we understand the ins and outs of that process, of that signaling process. Some of it happens at the endoplasmatic reticulum, some of it happens at the Golgi apparatus, and it involves some key transcription factor, which is called SREBP2, and that SREBP2 regulates gene expression of those genes shown here on the x-axis through a sterol regulating element. And if you knock out that gene, you expect all of those regulators of cholesterol biosynthesis and cholesterol uptake to go down. And then sort of beautifully, what you also see is that transcription factor needs to be activated by a protease, and that protease cleaves off that N-terminal piece that then ends up sort of entering the nucleus and regulating transcription. And also that knockout for that protease, which is S2P, leads to that downregulation of these cholesterol biosynthesis and uptake genes. And sort of, we even find sort of converse regulators, other regulators that negatively influence that process. One of them is INSIG1, which is actually also included in this cartoon. And GBF1 is again a gene that resides in the Golgi apparatus, and it's been described as a sort of intriguing candidate for regulating that process. So a lot of this actually makes sense. And again, I like the thought that by enlarging the panel a little bit, we can query a totally different area of biology here. And because these screens are genome-wide screens, they will contain that data anyway. With that, I'd like to close and conclude that we're very intrigued by this compressed perturb-seq, CROP-seq screening paradigm, because it allows us to squeeze very large screens into a very small number of cells and make them affordable and sort of doable. We're excited about targeted sequencing because it leads to a significant cost reduction, and at the same time increases the depth of transcriptomic data. And I should add here that this is something that we do in-house, but it's something that BD Bioscience is also willing to help you with. So there is actually a targeted sequencing sort of panel design and panel creation service that is very helpful. The main screen, if you like, that we conducted identified key factors involved in M1 macrophage polarization. And then I mentioned this, what I call a side screen here. This is a sort of, if you like, a side activity where we looked at cholesterol biosynthesis, and we recapitulated some of the known biology by sort of finding S2P, this protease, and the transcription factor as key regulators of cholesterol biosynthesis. And then finally, we're excited about AppSeq and CITE-seq because it enriches and complements single-cell RNA-seq data. Finally, I'd like to thank all the people involved. This is the entire Milia team. At Milia, this was spearheaded by Daniela Fleberger and Katarina Lalatovic. together, I should say, with Sumit Pava. And then I need to also thank my colleagues from BD Biosciences who supported this very generously and sort of both by providing the means to do it, but also sort of helped us get the internal support. And this is Janis Ulbrich and Valia Lopez Salmeron. So with that, I'm at the end. I'd be very happy to take questions. Sorry, Hasi, I cannot hear you.
Sorry, can you hear me now, Tillman?
Yes, I can.
Perfect. Sorry about that. So first of all, let me thank you for a great presentation. These compressed screens are definitely very impressive. And so, yes, thank you again for that. So we're now ready for the Q&A session. So please remember, if you haven't already, to submit your questions into the Q&A box, and hopefully we'll have time to address them all. So just looking at the box now, we already have a number of questions. So let's dig into those questions. So the first one, Tillman, is what are the requirements for a cell line to be used in this compressed CROP-seq screening workflow?
Well, I would say the one key requirement is that you need to have a dCas9 CRAB derivative. So you need to have a cell line in which you have a stable integration of the dCas9 CRAB transcriptional repressor. And the second key requirement is that you need to be able to infect cells at a very high infection efficiency or a very high multiplicity of infection with the lentivirus. And that is sometimes easier and sometimes more difficult. So if cells are quite refractory to lentiviral infection, then you will not be able to deliver multiple guide RNAs per cell, but you'll be able to deliver one or two, which is then not really helpful because the degree of compression, if you like, is not fantastic anymore.
Thank you. And can this CROP-seq workflow be extended to primary cells?
Yes, I mean, we do a lot of work in primary cells already, and we find it very useful because in primary cells, in contrast to these THP1 cells, you have a lot of heterogeneity built in. These cancer cell lines are so popular in part because they're so homogeneous. They grow like wheat, they're very easy to handle, and they're very homogeneous. Now, the heterogeneity that is inherent to biology is something that single-cell sequencing can capture really well. So we are very excited about using these technologies in primary cells. We've used primary T cells so far. Other cells, I would say, are under investigation. The one caveat that would apply is that in a primary cell, it's difficult to envisage this scale. So it's usually smaller scale screens in primary cells.
Great, thank you. So the next question, how do you design primers and which features are taken into consideration for designing the primers?
Yeah, that's a good question. I mean, you could turn to BD Bioscience and ask for their help because they have their own algorithm and they can do this really well. You could also look at that publication, which we've found very helpful. This is Daniel Schweifvogel from EMBL. In general, we like to record bulk RNA-seq data before we design primers, and we like to record it using protocols such as the quant-seq protocol from Lexogen, which are 3' focused. And so by doing so, we have a very good understanding of where transcription of a given transcript terminates in a cell. You need to know this in order to be able to amplify that end of the mRNA. That's probably the key requirement, sort of understanding where transcription stops. Other than that, there is sort of, I would say, I wouldn't call them generic, but there are some design rules that go into this that avoid sort of crosstalk between different primers that sort of allow you to target regions that are non-redundant and so on. But that, at my point of view, is sort of not rocket science anymore. It's really knowing where transcription terminates. That's the key piece of information that you need for this to succeed.
Makes sense. Thank you. Okay, so next question: how many guides are tolerated per cell, and what's the upper limit?
Yeah, that's an interesting one too. The first screen that was published with that paradigm, they didn't call it like this, was a screen published by Molly Gasparini, and she worked with a cell line called K562, and she managed, to my recollection, to get in more than 20 guide RNAs per cell. So that was sort of on the upper end of the scale. We've conducted several such screens, and I would say our experience is that, THP1, we got 5 guide RNAs per cell. We had done a similar experiment in a Jurkat cell where I believe we got to 13 guide RNAs per cell. So I would say the range that we're looking at is sort of anywhere from 3, 4, 5, all the way up to 20 to 25. That's the range.
Great, thank you. I mean, 20, that's quite amazing. And can you squeeze the experiment in even less cells? I mean, million cells, coming from 9 million to 1 million is already a great feat, but I mean, do you think you could go even less cells?
Yeah, I think less is difficult. Generally, the power to discover things in single cell sequencing is very strongly correlated with the number of cells that we sample. And in this particular experiment, we were at around 150 cells per gene that we perturbed. That is already, I would say, on the lower end. In fact, if I were to do this again, I would probably try to go a little bit higher just to be able to uncover a little more subtle phenotypes. If the phenotype is very strong, and for instance, the TLR4 knockout phenotype in that experiment was a very strong phenotype, you can uncover it from very few cells. But if the phenotype is a bit more subtle, it's helpful to have more cells in the experiment. So I would probably not go much lower than what we had done here.
Okay. All right, so the next question is, what would be the number of cells needed for a standard genome-wide screen versus the compressed approach?
Yeah, we calculated that you need approximately a standard genome-wide screen would target 20,000 human genes, and if you want to be at 100 or let's say 100 to 200 cells per guide RNA, I think a conventional sort of screen would be anywhere from 5 to 10 million. I projected in that slide deck, I said 9 million. It's again a bit dependent on how deep you want to go. And then the compressed screen would sort of reduce this by a factor of 5 or 5 to 10. That is kind of the gain that you have. And that gain is very significant because single cell sequencing is still an expensive technology, I would say.
Yeah, no, I agree. Although, I believe the costs have come down slightly with the newer NovaSeq X system. But yeah, for certainly when you're doing so many cells, which makes this compressed genome screen definitely more advantageous. So a few more questions here. So how does the Rhapsody technology or the workflow differ from other technologies?
Yeah, what we like about the Rhapsody particularly is the imaging piece. So we're able to scan at any stage the cells. So you essentially scan the little grid that separates the cells and the beads, and being able to see what's going on there, being able to see the cells as they enter the wells, being able to count them, gives us a very clear expectation of success or sort of lack of success, and allows us to predict whether it's actually worth spending a lot of money on that experiment. We've had cases where already at that stage we could see that something didn't work, and in that case we wouldn't spend an NovaSeq X run to sequence. So that, I would say, is, at my point of view, for us the key differentiator to other sequencing workflows. We really like the imaging piece.
Great, thank you. Yes, I think it does give a lot of confidence being able to see what you've actually captured, and especially again, even though this is a compressed screen, you're still working with a large number of cells, and ensuring that you have captured that does make the difference, I can imagine, from a confidence level. Yeah. Absolutely. Okay. So can this type of screen be used to study combinatorial effects?
Yeah, that's an interesting one too. We get that quite often when we speak about that paradigm. And I think the difficulty is, or what people sort of underestimate, is that the combinatorial space, if you look at a cell having 3, 4, or 5 guides, the combinatorial space just becomes enormous, meaning for any given combination of 3, you might find 1 or 2 cells or even no cells in that experiment, and that would be insufficient to call effects. So while it's sort of intuitive to think that you could capture combinatorial effects as well, you will usually not ever find at least 20 cells that have the same combination of 3 factors in them, which would be the minimal requirement to be able to do so. So usually not, or sort of really not.
Okay, great. So next question, could you also implement intracellular proteins in such a CRISPR screen?
Yeah, I mean, we're very interested in exploring this. We've, again, worked with Badia and team at BD, and we're looking at that technical solution as we speak. Obviously, protein is, transcriptome is sort of a proxy for protein, right? I think what people would, if there was no restrictions on technology, what people would actually love to do is look at all the proteins of a cell rather than all the mRNAs of a cell. So we have a lot of interest in trying this out. We'll see whether it's useful. There is still, at my point of view, there is still a restriction in that, even for cell surface markers, we now have site-seq panels covering, 150 genes, maybe 200 genes. For intracellular, the offering is even more limited. So the worry is a little bit that we will always be interested in the one protein for which no antibody has been sort of validated and made. But yes, we would be very keen on exploring this. Great.
So a few more questions here, and I think we've still got a little bit more time. So let me carry on. How many protein markers could you analyze in parallel in such a screen?
I would need to go back to the team and ask that question. It's in the order of magnitude of 150, if I recall correctly. So there is actually antibody panels that have been pre-designed and created for specific applications, particularly in the immune space, and we used one of those. So it's, I think the limit is really really lies in the reagents that are available, I would say, right? Because all of these antibodies need to be coupled to oligonucleotides, then the unbound oligo needs to be removed, you need quality control, then these antibodies need to be titrated, then mixed. So a lot of work goes into devising those antibody panels. So that's why they're growing that slowly, if you like, and that's why only few of them are available at the moment, I would say.
Mm-hmm. Okay. So here's a question here, if I'm reading this rightly. Have you used enzymes for bringing the cells in suspension, and did that have an impact on the protein analysis? So the sensitivity, I guess.
Yeah. Well, with these cancer cell lines, I believe we usually use trypsin to get them into the single cell suspension. We've worked with other model systems where sort of trypsin was not an option, and where you had to apply sometimes more gentle, sometimes harsher solutions. I would say so far we haven't encountered a cell that we could not sequence. So there is always a technical solution to this, but depending on the complexity of the cell type and the, let's say, the geometry, the morphology, sometimes you need to sort of try a few possible solutions to find one that works.
Okay. All right, so next question, I think we covered this, what is the maximum number of genes you could cover with a targeted panel? And does the Rhapsody allow WTA single cell RNA sequencing, which I could answer, but I will let you answer.
Yeah, that's a good question. That's actually another piece that is nice. So what the Rhapsody allows you to do is it allows you to do targeted sequencing and whole transcriptome sequencing one after the other, meaning you can take the same beads and you can process them first. I don't remember the order. There is a particular order that's actually recommended, but you could do targeted first and then whole transcriptome, and that would allow you to get sort of, on the one hand, the full picture, and on the other hand, the targeted picture from the same beads. What's the upper limit of the panel size? The original paper by Daniel Schreivogel suggested that a panel of up to 1,000 mRNAs could be targeted. We routinely do 500, and we've had selected cases where we've done 700 or 800. So I think that I cannot tell you precisely, but the limit is anywhere from 700 to 1,000 mRNAs targeted in one panel.
Mm-hmm. Great. Dropouts. How does the dropout rate in the targeted assay differ from the whole transcriptome assay?
Well, the whole transcriptome assay, I would say, has very little dropout. It will just miss out on a lot of lowly expressed genes for which you never see a read or see very few reads. With targeted sequencing, you increase that. However, there is a small set of mRNAs for which the algorithms don't find the right primer pair. I didn't point to this, but usually every panel, let's say every panel of 200 has a set of 3, 4, 5 mRNAs that for which primer design does not work. So there is a small sort of technical dropout. And then, I would say it's really the very lowly expressed genes that sometimes you cannot capture. This is sort of, if you recall from this tiered experiment that had the 7 expression level tiers. From that tier 1, you sometimes struggle. This is the tier that's closest to the bottom, if you like. But all other tiers we routinely manage to amplify.
Okay, thank you. So I've got one more question for you before we wrap up, and I think this is a good question to finish off with, is what steps of a CRISPR screen do you offer as a service?
Yeah, we like to perform these screens as an end-to-end offering, really. So meaning people come to us, they develop the assay with us, they say, this is the cellular model we have in mind. Sometimes they have it physically, then it can be transferred to us. Sometimes we source the model. We then develop the assay. We usually spend a significant amount of time developing the assay. And then once we all agree that the assay is fully developed, we run the screen and we do library prep, sequencing, and bioinformatics in-house. So we really think it's the best if we sort of own the entire process from beginning to end, so that the quality is what we would like to have and we would like to achieve. So it's really the whole process from assay development all the way to data analysis that we offer to clients.
That's fantastic. And I think that gives a lot of confidence for people who are new to this, and to have that support is very valuable. So yeah, if anybody is interested, definitely reach out to Tilman. Okay, so I'd like to thank you, Tilman. That is the end of our Q&A and the webinar for today. I'd like to thank you all who have joined us for the questions and also for your engagement. If you'd like to get your questions, so we will respond to the questions that we haven't answered. If you have any other questions or queries, please reach out to us at SCOMIX, which is single cell omics, so SCOMIXBD.com. If you have questions for Tillman, definitely reach out to the Milia team. And so that just leaves me once again to thank you, Tillman, for a fantastic presentation. And I wish you all a good day. Thank you.